This video is going to cover translation. You should have received a codon chart, so it has a big circle on one side and it has a square on the other with a bunch of letters and some names of amino acids. You are going to need that for this video, and you are going to need that for when you're doing your practice and your exit assessment, so make sure that you have that before you begin. So let's take a look at translation and what it actually is. So what we're going to do is we're going to start with a single strand of RNA, and we're going to translate it into an amino acid chain. And when we put amino acids together, we know that means we make proteins. So you can think of this as if you're translating something in a language, you're taking it first from something that does not make sense to you into your own language. So we're going to take just these letters from RNA. When you just look at them, it doesn't really make a lot of sense. And we're going to translate it. We're going to put it into something that we do understand, which is amino acids. And then we look at the amino acids that we have, and then we'll be able to identify our protein. So a key idea here is that if you are given DNA and you are asked to translate, not transcribe like you did before, but translate, it does need to be in the form of RNA. So if you're given the DNA, you need to convert it to RNA first before you can translate. So you need to transcribe and then translate. So you read about codons in your reading guide, but just to recall that a codon is what we refer to as 3 bases in a row that are grouped together, and then those 3 bases will correspond to a specific amino acid, and that's what you're going to see on that chart that I just spoke about. And those amino acids can be found on that chart. So we'll learn how to translate today by looking at and using our codons and using that chart. So before we look at just a giant chunk of DNA, let's just look at a few individual codons. So you'll see 2 sides of your chart. Let's start on the side that has the circle. With the one on the circle, you are going to work from the inside out. So you're going to start with whatever your first letter is. It's C, so you're going to find the C that's in the middle, and then you're going to work your way out. So from where that C is, you're going to find a G. From the G, you're going to keep moving out to look at your last letter, which is an A. And then if you look on the chart, that will give us the amino acid arginine. So if we have that amino acid, or if we have that codon, then the amino acid that corresponds with it is arginine. So next one, AGC. So once again, you're going to start in the middle. Start with the A, moving out to the G, out one more time to the C, and what you should see is serine. So here's one. Try it on the circle side on your own, CAG. Pause the video, take a look at it, and then come back and see if your answer is correct. So what you should get with that is glutamate. Now I'm going to ask you to flip to the other side. You want to be comfortable with both. In terms of what you use on your assessments and what you use on your test, it does not matter which side you use. However, want to make sure that you are familiar with both sides, because when you take your air test at the end of the year, you want to make sure that you're able to use both sides, because both of those have appeared on the test. So it's okay if you're more comfortable with one than the other, but you want to make sure that you are familiar with both and you can use both to be able to read them. So if you look at the other side, your first nucleotide is on the left-hand side. So that's the first thing you're going to locate. And then you're going to find which row it matches up with. After that, you're going to look in that row and you're going to go across the top to find out which column your letter is in. And then from there, you can go to the right side to find out where your third letter is, to see which one it is. On the side with the square, your amino acids are abbreviated, so it's going to look a little bit different than they do on the other side. I'm okay with you either writing the abbreviations or writing the full word. It does not matter either way. So let's take a look at the first example that we're going to use on the side with the square. So UUG, so looking on the left-hand side, you're going to locate U, finding that row that U is in. Then going up to the top, find the column that corresponds with that, and then find where the column and the row come together. And then within that box, you should see UGG as an option, and then you'll see leucine written there. And I believe it's abbreviated LEU on that one. So let's take a look at another one before you try one on your own. AUA, so we'll find the row for A, go to the top, find the column for U, find where they meet, and then within that box we should see AUA, which is going to give us isoleucine. Now, same thing that we did before, take a look. Here's your next one. On the square side, pause the video, see what amino acid you get, and come back in just a second. So what you should get in this case is histidine. Now, there's two others that you will see quite often. You have AUG. And on the square side, you will see it's written as MET, but it also says START. So that's methionine, but it's referred to as the START codon. On your circle side, it does not say START, so what I would like for you to do is write START next to that. So find AUG and you'll see methionine. You're going to write down START next to that. So you know that this is the START codon. This is what every protein sequence is going to start with. It's the way that the ribosome knows that this is the beginning of a protein. And we'll talk later on about what that actually looks like and where the ribosome comes into play. But for right now, just know that that is when we have the start of a protein. Now, if we have a start, we also have a stop. So this is just one example. There are multiple stop codons, so you'll see that on the front side and on the back. The stop codon tells us that this is the end of the protein. So you'll have a start, tells you where the beginning of the protein is, then you're going to have some amino acids thrown in there, and then you have a stop at the end. So now let's take a look at what this is going to look like without just individual codons, if we actually have a piece of RNA. So translate this piece of RNA. The first thing that I'm going to encourage you to do is take this strand and actually break it up into codons so you can keep track of where you are. Because what we don't want to happen is we don't want to skip a letter. We don't want to do a letter twice. We'll talk later about mutations and why this is bad. But you want to make sure that you're using all the letters, you're using them in order, and you're not leaving any out. So what I like to do is I like to count 1, 2, 3 letters and draw a line. 1, 2, 3 letters, draw a line. 1, 2, 3 letters, draw a line, and then that's the end. Now I have a clear way of seeing, okay, here's my first codon, here's my second codon, my third codon is ACA, my fourth codon is UAA. So it's really clear to you which codons are which and which letters match up with which one. It's going to make it a lot easier when you have longer strands of DNA. I know this one's not too long, but when you have longer strands of DNA or RNA, it just helps you out, keeps everything a little bit more organized. So we're going to do the same thing. Feel free to use whichever side of your chart that you want to in this situation, but we're going to start out by looking at AUG. So if you remember from our last slide, AUG is methionine, which is our start codon. So that's the first one you're going to write down. This is our translation. This is the part that we have that makes sense. It's something that we can actually read. If you go to CCC on your codon chart, so either working from the middle out on your round side or left, then top, then right on your square, if you match up CCC, that's going to give you the amino acid proline. So you're going to write proline down. So once again, I said if you are working on that square side, abbreviations are just fine as well. Don't make up your own abbreviations though, because you'd be surprised as to how similar some of them are. So if you don't have the abbreviations, please write out the entire word. Then ACA, if we are looking for that one, that is threonine. And then our last one, UAA, is a stop codon. So we have our start codon, and then we have our amino acids, and then we have our stop. So just know that proteins are not going to be 2 amino acids long. They are going to have more amino acids that correspond with them. But this is just kind of an example to get us started here. All right, last example. I'm going to have you try this one on your own. So it's going to pull in some stuff that you have already done. But we're translating this piece of DNA. So one thing I want to point out before you get started is this is DNA and we need RNA. One way that you're going to quickly realize that what you have doesn't match is if you look on your circle side or you look on your square side, either one, you are not going to see any T's. So remember that DNA has T's and RNA has U's. So if you try and translate this piece directly, you're not going to find anything that corresponds to TAT. That's not going to match anything. So make sure that you transcribe this piece first, and then you're going to go back and translate it. So hit pause. Try this on your own. Make sure that you are familiar with what you are doing. If you're willing to try it, flip your chart over. Try the side that you're not super comfortable with, and we'll go over this in just a second. So go ahead, hit pause. All right, so here is your RNA sequence that came from this piece of DNA. So remember, my T's still go to A's, but my A's go to U, and my C's and my G's are going to stay the same. So here is your piece. And then you should have isoleucine for your AUA, alanine for GCC, tyrosine for UAC, and glutamic acid for GAG. All right. So if you have any questions, please let me know. If you are ready to do a few more practice problems, give a few more of those practice problems a shot, and good luck.